Okay, we can probably get started. My name is Joe Gleason, and I have the pleasure of introducing today's speaker, Jerold Chun and Jude Onya from Neurocrine. Jerold is a distinguished scholar at Neurocrine, and Jude is the chief scientific officer. Neurocrine, if you don't know, is a major biopharmaceutical company here in San Diego, and we're so thrilled that they're here in our neighborhood. And I think there's lots of opportunities to connect. Neurocrine is basically what it is: neuro and endocrine. Trying to think of therapeutic opportunities, and they're undergoing somewhat of a transformation right now. And I think it creates opportunities for synergy and for collaboration. So Jerold will be speaking today. And Jerold, you probably know, has been in San Diego for a very long time and has worn multiple hats. He graduated from the University of Hawaii and had an MD PhD at Stanford in neuroscience, working with Carla Schatz, and then was at the Whitehead for postdoctoral fellowship in developmental and molecular biology. He was a professor here in pharmacology for many years. And he was a postdoctoral fellow with Rudy Janisch and David Baltimore. And he rose through the ranks, assistant associate and full professor in pharmacology here at UCSD, and was for a time the director of the neuroscience graduate program. He was at Merck for some number of years, was at Scripps Research Institute, and at Sanford Burnham Prebys. And he has been a real mainstay in the neuroscience, in the molecular neuroscience field, I should say, studying many aspects of that are both in the area of at the pharmacology neuroscience interface, working along GPCRs and molecular mechanisms of disease, including mosaicism and Alzheimer's disease. And recently has joined Neurocrine to help usher in a new wave of therapeutics. Jude Onya, who's joining today, is the Chief Scientific Officer at Neurocrine since November 2021 and leads drug discovery and non-clinical developmental teams responsible for bolstering and advancing the company's pipeline of therapeutic candidates. In the beginning of 2023, Dr. Onya joined Voyager Therapeutics Board of Directors. He has more than 25 years of experience in pharmaceutical industry. He's the former vice president of biotechnology and discovery research at Eli Lilly. And at Lilly, he contributed to the discovery and advancement of more than 60 clinical candidates across multiple therapeutic areas, which led to 7 approved medicines, and was responsible for more than 50 pre-candidate programs across multiple therapeutic areas, with a bachelor's degree from forest biology from State University of New York, SUNY, and a PhD in cell and molecular biology from SUNY Health Sciences, both in Syracuse. Okay, all yours. We're going to do kind of something new today, which will be some part of an introduction to what Neurocrine is doing, and then open it up to Q&A. So please keep your questions and think about what kinds of things you want to ask.
Great, thank you very much, Joe. Yeah, it's a pleasure to be back to UCSD, and this is really my longtime home. And now there's, is this too loud? It sounds really loud.
You put it down a little bit.
Is that better? Yeah, better. Yeah, we've now entered sort of a new era when you think about the potential for neuroscience therapeutics. And Neurocrine, a local company here, now has, I think, the potential to really make a difference, both from the therapeutic standpoint and as a member of the San Diego community. And so our role today and purpose today is to introduce you to Neurocrine. And that begins both with myself and then, as you heard, Jude Onya. And I know the camera can't see him, but we'll try to track him over there. But he's here with us today as well, and will be available for questions as we go through the presentation. So what is Neurocrine? Neurocrine is a company that was in fact founded by some very famous scientists that one of whom was here in the area, and that's Wiley Vale back in 1992. And unfortunately, Wiley left us too soon, but many of the projects that Wiley worked on in the endocrine space are still being carried on therapeutically in Neurocrine today. And then also Larry Steinman is one of the other founders. He's a very prominent multiple sclerosis neurologist, and he was in fact one of my teachers when I was a medical student. And so both Wiley and Larry were the founders. And then a lot's happened in the interim. Neurocrine underwent an IPO in 1996. And then after many, many challenges that are not unique to Neurocrine, but to their president and all of biotech and pharma, they did manage to achieve a real success, which is the approval in 2017 of a medicine called Ingrezza or valbenazine. And we'll spend a little bit of time shortly to look at that. It's indicated for a disorder called tardive dyskinesia. We'll talk about that in a bit. And this was discovered and developed at Neurocrine. So the company is focused on discovery and development of novel therapeutics for CNS and endocrine diseases, as the name reflects. And it's now approximately 1,350 employees and growing. So it's going through a growth phase. And it involves both the sales and commercial side, the R&D side that we'll be talking about today, as well as the very important clinical and regulatory side of the company. It's located here in San Diego. It has global and regional partnerships. And some of the insignias you see here are companies that Neurocrine's interacted with over the years or currently. Here's pictures of the board or primary management committee members. I won't take you through all of them, but I wanted to highlight Kevin Gorman, who's the chief executive officer. And Kevin has been there 15 years now, and the company really reflects a lot of his values and ethos. Kind of a story on the side. One of the first interactions I had with Kevin was meeting him in Hawaii, and both of us were in our aloha shirts, and it was sort of a sign that it was meant to be to work together. Many of the others have ties to the San Diego area, and you might have interacted with some of them over the years. And then, of course, Jude, who guides our R&D and oversees things. And so here's our leadership chart. And so Jude oversees both the research side and preclinical and CMC sides. And I help in that regard to provide some neuroscience expertise, particularly if we can bring it in from academia, some of the cutting edge things, for example, that are going on here at UCSD. The gang is really an exceptional group. I'll just highlight some of them just because you might know some of them, but also to illustrate both the expertise and the diversity of the group. Sancheng Na is the neuroimmunology lead, so it's basically the department head of that group. Brings in many, many years of experience in the immunology space. Tom Kornacek, who came from both Merck and Biogen, leads our in vivo translational pharmacology. Fiona Scott leads the endocrine piece, and she came from both Receptos and Celgene. I'm going to pass over to Nicole for a second, but note that Steve Perry also brings in many, many years of experience at Neurocrine, as does Gordon, who's our VP of preclinical, and Jay, who covers the CMC aspects. And I'll return to Nicole now, because Nicole, in fact, was a graduate of UCSD, received her PhD in chemistry here a while back, and she's now our head of chemistry. So we have this great group of folks, and they're guiding us in getting new products over the finish line. The way we're doing this is through therapeutic areas, and we have 4 therapeutic areas that cover a wide swath of neuroscience. There's the neurology side, psychiatry side, endocrinology, and neuroimmunology. Within each of the therapeutic areas, we cover the research, clinical, business development, commercial, chemistry, biologics, and of course, the important regulatory piece. And so that's shared amongst the different therapeutic areas. And we are using these in order to help us to select new projects and also to make progress in new areas. The general strategy is shown with the diagram to the right. And so this ties into many of the things going on at UCSD, where one wants a deep understanding of disease that ties into unmet medical needs, and that we can also achieve clinical and genetic validation for anything that we go after. Make sure we have relevant endpoints that are translational, capitalize on shared mechanisms, explore multi-target drug combinations, and then approach it in a modality agnostic strategy. And that's really important because it's not just small molecules today, it's actually far more than that, and that's shown in the next slide, where in fact Neurocrine has access to all of these different technological platforms. Small molecule side, which is important and is a part of what we do, but in addition, there's the peptide piece, there's the antibody piece, which is, of course, a fantastically interesting area with all the things you can do there, and the gene therapy piece, which is one of the hottest areas out there and still in its infancy in many ways, but has perhaps some of the greatest potential, especially to really curing familial forms of a number of genetic diseases. And so it's this combination that is being pursued through the therapeutic areas, and we'll select the approach that is most appropriate and should have the highest likelihood of success as we go along. So what has Neurocrine done? Well, they have a number of approved drugs. The one that we'll talk about most today is Ingrezza, and I'll come back to that particular agent, but they also have Ongentys, which is opicapone. It's a COMT inhibitor used for adjunctive therapy in Parkinson's disease. And then there are two other agents, Orlissa and Orhan, and Orlissa is a GnRH receptor antagonist. And then it's also in combination with progesterone and estradiol. These are for the treatment of different gynecological conditions, endometriosis and fibroid pain and bleeding. However, there are a number of other agents that are coming down the pipe, and this is just a snapshot of the clinical pipeline, just to give you a taste for what's happening in the different therapeutic areas. In neurology, there's valbenazine itself that's being looked at in chorea and Huntington disease, and there was just a paper published last week or so in Lancet that detailed efficacy in that setting. There's dyskinetic cerebral palsy also being examined. And then the next three relate to various ion channel types of approaches to look at epilepsies. In the neuroendocrine space, there's crinecerfont, which is being looked at for congenital adrenal hyperplasia in adults, as well as children and adolescents. And this is a CRF1 antagonist program. And then in neuropsychiatry, also valbenazine, and then a number of other agents that are being looked at various disorders, major depressive disorders, cognitive impairment, schizophrenia, and so forth. So these are all at clinical stages and represent what's coming down the pipe. And then what we'll be doing in the coming years is to fill that pipeline with new things. So just to give you a taste for what Ingrezza involves and some of the biology behind valbenazine. So this is the treatment of tardive dyskinesia, which would seem to be something that is not so prevalent. And yet, as one goes to look at it more closely, it's maybe surprisingly common. It's a movement disorder caused by prolonged use of antipsychotics and anti-nausea medications and probably other agents as well. And it results in uncontrolled, abnormal and repetitive movements. So motor symptoms that have a negative impact on things like job performance, kind of self-worth, and it results in isolation of these individuals. And so this is something that can, if you can manage it, can actually change the way that individual lives. So how does valbenazine work? It's actually a VMAT2 or vesicular monoamine transporter 2 pathway inhibitor. And it's been found that it can treat TD or tardive dyskinesia. And if we look at that schematic diagram to the right of this synaptic terminal, we have a presynaptic terminal that's dopaminergic, but it could be any number of other monoamines, serotonin, histamine, epinephrine, norepinephrine, depending upon where one looks. And VMAT2 is shown as this transporter that basically loads up those vesicles so that it can be released, the transmitters can be released in the synaptic cleft and then produces changes. So by inhibiting it, you can prevent the vesicles from being loaded up with these signals, and that's the basis of the approach. And it's been found to be effective in that tardive dyskinesia, and it's now being looked at in that chorea and Huntington's disease, some aspects of schizophrenia, as well as the dyskinetic cerebral palsy condition. There are next generation molecules being produced, and Neurocrine really has a leadership position in this particular space. Just to give you a further taste of that market, there's an estimated 600,000 individuals in the US that suffer from this disorder. And that reflects the increase in antipsychotic prescriptions that has been going up in the US. And many of these individuals are not diagnosed and, of course, not treated. And so that provides an opportunity to get them exposed to an actual efficacious agent. So Neurocrine is well positioned for the coming years. There's Ingrezza. In 2023, the sales guidance is somewhere in the 1.6 to 1.7 billion, and that's related to that patient population. But in addition, the robust pipeline that's being produced right now through Jude's visions will fill in new agents, and that's certainly one of the things we'd love to talk to you all more about, where there are opportunities to, bring in programs that could be added to that effort. And the strong financial position means that Neurocrine will be here to stay for the foreseeable future. That includes a new campus that's being constructed, and some of you that live out past 56 may have seen this there. This is right next to Canyon Crest Academy. It's going to be at least a 4, potentially a 5 building site. And R&D, if all goes well, will be moving in there in 2025. So we're all excited to have some new digs, and this should also give us room for expansion as we go along. Some of the external innovation and acceleration activities come from working with companies, and these are just kind of a taste of some of the commercial relationships that Neurocrine currently has that relate to both the companies, the targets, as well as the agents. And so you can see there's a pretty wide range of things that have been connected through these companies, either through licensing deals or in some cases a bit more, such as purchasing parts or the full company. The one I'd like to highlight towards the bottom is Voyager, which is a gene therapy company out in Boston. And this relationship is one that's going to be central to the success of Neurocrine in that gene therapy space. The current projects that are being pushed quite hard are GBA1, glucocerebrosidase, and that's in Parkinson's, maybe also Gaucher's, as well as FA and Friedreich's ataxia, and there's some other targets being examined as well. But it offers, I think, a glimpse of the future, that as the gene therapy piece becomes more and more viable, that many of the genetic diseases can be approached in a really rational way and successful way. So what do we hope to do with the various academic groups? And it's something that I'm hoping we can leverage here, which would involve a number of different interactions. We have an active internship program that involves students as well as others at different phases of their careers that come in and spend anywhere from 6 to 10 weeks within Neurocrine. And that program enrolls every year around this time. And so we've actually started it. So the next available slots would be for the coming year. There's also a postdoctoral fellowship program that involves postdoctoral work, but working within some of the programs that we have ongoing at Neurocrine. And then, of course, scientist-driven interactions that would involve support for labs here at Neurocrine, or perhaps support for postdoctoral fellows within UCSD that would be supported by Neurocrine. And then the possibility of future employment at Neurocrine. As Neurocrine grows, it will need more skilled and exceptional scientists. So that's absolutely a place where we'd love to draw from from UCSD, as we've done with our head of chemistry, Nicole Harriet. And lastly, the intellectual property piece, which is very important, making sure that we're able to access some of the most interesting science and have it protected. And the areas that are open are many. Neuroscience, of course, but chemistry, molecular biology and biochemistry, the bioinformatics, computer science, pharmacology and immunology, and then the clinical medicine side where we are always looking for good clinicians or clinician scientists that can contribute to the efforts. So that brings me to the end of the formal presentation here. And both Jude and I would be happy to entertain questions. And want to thank you for joining us here. And if you have questions that can be sent in through the chat, we can address that as well. But we'd love to work with you more and have some fun times and making new neuroscience therapeutics. And so with that, let me open this up for questions. Go ahead, don't be bashful. It's and I don't know how to access the Zoom stuff, so yeah. Okay, great. Please. Oh. Of course, man. No, don't be ridiculous. Yeah, you're a star here, so it's this Abe's famous, so. No, just to tell the rest of you. So, yeah, my lab started in BSB over there many moons ago, and Abe was this intrepid soul who was working under dire conditions and emerged like this flower to blossom into something.
Okay. I feel like I started hearing 20 years ago that big pharma had moved away from neuroscience, right? And whereas the portfolio you're showing is kind of speaks in an opposite way. Do you see yourself as an outlier, sort of one of the pharmaceutical companies still in neuroscience? Do you think that story has evolved a little bit? But a lot of people in the academic space that still tell that story.
Oh yeah, well, it's true.
Yeah.
And it depends at what point you look at it. So back in the 90s, everyone cut neuroscience. And the most famous one was Genentech. And it was because it's hard. It takes you a long time. And even when you think you know what's going on, you don't. And then you go in there and say, hey, I got it figured out, and bad things happen, or it just doesn't work. And compared to cancer, where the safety is a much shorter period, and if something grows or doesn't, so the endpoints are easier. And yet, if you talk about medical need, there's never been a greater medical need for neuroscience therapeutics. Whether you're talking about neurodegenerative diseases or all the things that affect people's psyche, et cetera, et cetera. And then you can extend it even further, which is the brain is tied into every part of your body. So when you look at it that way, man, there's huge need that needs to be met. So our company is going back into it. Clearly, we have a few that are, and I think maybe the most prominent ones include Biogen, which has a big footprint in there. I'd say Regeneron, but they took a rather interesting sidestep by going the immunological approach to it, have been very successful. And then, of course, Neurocrine, and that our stated goal is to become the best neuroscience company. And so that's the plan. Now, how we do it, I think what Jude's put in place here is pretty remarkable in that we have these different platforms that can be accessed. It's not just small molecules anymore. So that opens up a whole nother set of potential approaches that we can get to. Now, do we have the runway to do it? I think the answer is yes, but you burn through a lot very quickly. So that's why we have to stay focused for a bit, see if we can get some winners. So that's the plan. Jude, yep, please.
I hope the rest of the big pharma stay out. Yeah, no, I was saying if I should add anything, I hope the big pharma they stay out. The less the competition, the better for us. But that said, as Jerold pointed out, we're really building to be a leader in the space. A lot has happened in the space. One is really that we're getting to really better understand cellular molecular pathways in the brain. The second is tools tools that actually allow you to go after targets in the brain as toward these biomarkers. And frankly, the agency is showing increasing willingness to approve drugs in the space with biomarker-driven approaches. And so bringing all that together, we see an incredible opportunity, and we're really looking for talent to help us win in the space.
Ruben. Yes.
I have a weird question about Parkinson related target dementia. And in some ways, it's opposite biologically, but they'll also manifest itself as, and maybe we call it a treatment of the adult brain. So can you speculate on the applicability of these agents against the Parkinson related?
I think any type of motor system that would involve that VMAT2 pathway, and Parkinson's fits in there because VMAT2 is expressed within the midbrain and substantia nigra and the VTA. So I think there is that possibility to access it. Now, it may be that if a patient is too far progressed, too much neurodegeneration in that area, you have less opportunity to. So it might be better for a less progressed PD patient, but certainly the biology supports some functionality there, and that needs to be examined.
In this case, it will be with like how it will be administered.
That remains to be seen. I mean, there's nothing approved in that space right now, so yeah, it has to be examined further. But it's an interesting thought. But it certainly is in the right place to do things. And it may be that different formulations or even next generation types of agents might be better suited for that. But that's for the future. Sure.
Can you give some examples of how Neurocrine sees collaborations with labs around La Jolla? Maybe some tangible examples, because I think what could we run the risk that Neurocrine could be viewed as a place that could recruit postdocs, maybe as a negative. So how can we turn that sense into not right? I mean, because postdocs shortage. How can we make this positive for standing alone? What kind of collaborations, what kinds of interactions would be possible?
I'm not going to answer the question, but I use the opportunity to tell you what an honor it is for Neurocrine to attract Jerold. This is part of Jerold's role, is not only to help us think about the strategy and the science and driving the programs, but frankly, to help us also think about how we build academic networks. And so this visit today is very, very timely. And so to your question, this is part of the things that Jerold is helping us think about. How do we do it well, okay, as a partner? I have my thoughts, but I'm going to shut up and I'm going to let him take the question.
Yeah, and we're all in the same boat, and it's a combination of, we want to drive the academic science to expand knowledge. Everyone that's in academia wants to do that. It's fantastically important. We also want to support the postdocs who are then going out into whatever that next phase of life is. And one of the challenges that I think everyone faces here in San Diego is looking at the job market and where folks go. And that begins with the graduate programs. And so I've been affiliated with both neuroscience and BMS for 30 years. And we've seen a huge shift in the interests of the graduating students, whereas most used to go into academic postdocs, most now do not. And part of that, I think, is economics, and part of that is lifestyle. And yeah, what I think the Neurocrine relationship could do is to provide another alternative to be able to both do really interesting science, but also have these contributions that you could call quasi-academic, but clearly would be trying to produce new therapeutics. So this is the type of thing we'd love to see brought about. And some of that, maybe there are postdoctoral positions within UCSD, within Joe's lab, for example, that then tie in more closely with the groups that I mentioned in our R&D. So that way you have the cutting edge technologies, they're aware of what's going on in the literature, but they also bring that into the picture. Because as many of you know, when you go into industry, oftentimes they're siloed. And many companies don't publish, or they publish in a very limited way. They're not open to what's out there on the horizon. And some of that's just practical, right? You can't do anything. You have to be critical path. But maybe there's another way to do it that will get everyone to the right point faster and in a better way. That's the hope. And I think the real pie in the sky is that at some point in the future, if Neurocrine becomes very successful, then you have sort of a Bell Labs of neuroscience drug discovery. That would be the kind of pipe dream. But even if you got partway there, it'd still be a fun adventure. And ultimately, it's patients that matter. So what can we actually get there for patients that will help them? You know, and for all of us that have ever looked at drug discovery, you know, that's the ultimate driver, because you can go make money in lots of ways, or you can write more grants, and you can do all of these types of things. There's lots of things you can do, but the patients are out there and they are suffering, and there's a real need for some new therapeutics. So why not contribute in that way? So those are some of the general kind of thoughts I have about it, and a lot that I think will, with Abe, the blossom analogy, to bring it out will be from working closely with you all. So, and what I absolutely want to stress is this is a collaborative and friendly kind of interaction that also will date back to, for those of you that I haven't met, I've literally been here for 3 decades in the San Diego area, which is a little scary to think about. But it just means that we have a lot of ties and a lot of friendships that also can help to smooth the way. So you know that there's not some evil ogre monster waiting, in some dark room in a company somewhere. It's not like that at all.
No, the only thing I would add is I'm one of the few who, from onset and graduating with my PhD, it was clear to me I couldn't survive in academia. It was pretty clear, and I wasn't sure what to do. And one of my mentors recommended an industry postdoc, and I went straight from Syracuse to Indianapolis to work as a postdoc at Lilly, and what ended up being a 25-year career. I cited for a specific reason, because while I was at Lilly in Indianapolis, we had this robust relationship between the IU School of Medicine and Lilly, where postdocs, we had robust postdoc programs where you funded postdocs, either in the university or individuals who wanted industry experience was also funded to come over at Lilly and do their postdoc. So, Joe, I see that same kind of thinking, opportunity to, do it like never before, where we satisfy the needs of folks who are graduating and thinking about whether they want to do industry or whether they want to stay in academia, and we can figure ways to fund these either there or there. Okay.
And it's a very topical subject, and all of you are probably familiar with Altos Labs that came up, and they've taken another slant on the puzzle. So I think there's lots of different ways you can approach this, and there are pluses and minuses to them all. But the bottom line is that lots of opportunity. It's a great time to be in science. So take advantage of it for those of you that are at that stage of your career, and those of you that are faculty. We'd love to talk to you more about what you're up to, and let's see if we can do things together.
Jerold, this is Terry Gasterland. I'm in
Hey,
Zoom.
Terry, I saw you.
Hi.
I saw your name zip in there. So how are
Yeah.
you?
Good. I'm in Zoom land, and I put a couple questions into the chat, but I'll just go ahead and pose them. I'm the director now of the Bioinformatics and Systems Biology program. Kevin Corbett is director of the BMS, Biomedical Sciences program, and students from both of those programs are attending the seminar. And we've got 100 students in BISB getting their PhDs. We have 350 bioinformatics undergraduates on campus. Across the computer science, bioengineering, chemistry, and biology departments. And we basically the undergrads and the grad students are really interested in 3-month summer internships. For our grad students, those internships can be as much as they could be pre-doctoral situations. IBM, Almaden has a really strong track record of taking a pre-doc, and then a student shapes their PhD research around an open problem that they found at IBM. That happens in computer science as an example. Does Neurocrine have a formal internship or scholarship program that the students could tap into?
Yes, there is a formal program. I would say it's not at this point tailored for bioinformaticians, but I would love to get that piece going. So we should talk. We should talk.
Yeah. And then related is, does Neurocrine have prion diseases on the radar? I guess that's not related. That's unrelated. For example, sporadic CJD or some of the familial forms of the disease.
We're aware.
Okay. So nothing specific. Okay.
But Terry, it's good. So kind of a sidebar for, when I was working at Merck many moons ago, I tried to hire Terry in, but she was too smart. She was too smart. So
well, then we co-advised a student together, and that was great.
Good to see you. Let's talk some more.
Yes.
What are the skill sets you think, maybe you could repeat the question too, because some people can't hear. What are the skill sets you think the trainees need to go into pharmaceutical if they're coming out of a master's or a PhD program?
Well, I think Jude would be able to nail that down for what I think everyone would say. You want someone who has the best scientific training possible, so they know what good science is. And that includes all of the aspects of reproducibility and rigor. And then, of course, any of the cutting edge technologies that would be amenable to pharmaceutical use. But, as you know, technology changes so fast. So, you want to pick things that actually will make a difference. And beyond that, it turns out that the skill sets that you develop in industry, while they're science, they are different in terms of the philosophy, in terms of critical path, in terms of what you prioritize. In the basic sciences or academic sciences, you can pretty much follow your nose to wherever it goes. But that's not so true in industry, especially when you have deadlines and other pressures where you have to get things accomplished. So that's just a quick rundown of some of the things. Jude, any things that you've seen? Yeah.
Not much to add. I will re-echo a couple of points. As Jerold points out, at the base of it, of course, is excellence in the sciences itself. I think it's an attribute that serves you well, whether you're in academia or whether you're in industry. But it's not just that. It's the edge. Having a differentiated edge in whatever you do, having the excitement and passion to really want to do what you do. And the third piece I will emphasize is the execution piece. It's not enough to want to do stuff. It's really being driven to get things done and executed and finished. When you come to industry, the last three things I was mentioning become important because it's really about getting things to the patient fast. And the science is good, but you have to be passionate. You have to want to execute to get to a timeline because the patients are waiting, okay? And it's also a pretty competitive space. So what makes you successful in the industry? I would say, I think it's essentially similar in academia. Besides the science and the things I've mentioned would be, frankly, teamwork, okay? It may well be, I think it's far more emphasized in industry. I have a friend that always draws this analogy about industry and academia. He says, industry is like traveling on a cruise ship, and academia is traveling on a speedboat. You can move fast, you can be lean, you can move. In industry, it's really a cruise ship. You have a lot of folks working together to really get that thing to where it needs to go. So the teamwork is one. I will also emphasize communication, and I want to take a detour on communication. It's not enough to be able to publish. I think oftentimes there's a notion that in academia you don't have to publish, in industry you don't have to publish. While publication is not a key measure, you still have to publish. You still have to present. You still have to, again, you have the skills to deliver, convince leadership to make investments in the things you want to do. So the communication is equally important. And the last piece on the communication I would emphasize is around soft skills. Again, I tend to define the soft skills of being able to say what you're saying in a way that everybody will hear you and everybody would want to participate in it. And I think it's a skill set that I've seen folks in industry who hone it very well, do very well in it. And I think the last one I would say is maybe mentoring. And I think it's through, whether you're in academia or in industry, it's not enough for others to mentor you. And the expectation is that you also participate in mentoring others. It's part of the culture of training, being trained and training others.
Great, great. Me, yep. Other questions? Please.
I have a very naive question. I'm not very familiar with how pharmaceutical companies operate. I'm curious how you guys allocate resources to target identification and drug development separately. And I can imagine if once you identify a target, you then want to shift resources to developing a drug against that target. And are you often kind of shifting people between those two roles? Or do you have separate teams for each of those roles? And how do you think about it?
Can you repeat the question just because?
Right, yeah. So the question is how you allocate resources for sort of target discovery versus development after when you start making compounds and going after that particular target. And a lot of the target identification will come from even the non-competitive space to see things that might make sense, and it might be a genetic link, for example, or it might be your competitor that's already made a drug against it. There's any number of ways that point to a validated target. If you're convinced the target's worth pursuing, then there's kind of a multifaceted approach to going after it that involves biologists, chemists, could be protein chemists or engineers, it could be immunologists, just depends on the approach that you use. So the short answer is we would assemble a group to best prosecute that project, but it'll depend on what the actual substance is that we're going after. So it'll vary from project to project. Ruben. Yeah.
I have a question about your internal kind of project strategy, and part of the question is there's competition for there's relugolics and there's amygdalics, and the relo has a CBT of something like 0.3 nanomolar, while Ella is 1.5, and then half-life is relo is something like 50 hours versus 46 hours. So then, in that sense, it may be interesting for you to develop a project which fixes those parameters within the same space, but it's kind of cannibalistic with respect to your previous product. So do you pursue those kind of improvements or dealing with the competition? Currently, relugolics in that sense wins a little bit in terms of market, no fun, but still.
All right, so Ruben is pointing out that so we have products that work in a certain way, and there's some distinguishing factors related to their KDs, and whether one can go after something that's more optimized at the potential expense of cannibalizing your product if you do that. And I think the answer is that if there's an opportunity from the patent standpoint and the market standpoint to do that, then yeah, then it might be interesting. But you're not going to go in there and shoot yourself in the foot if you can avoid it. So that's kind of a short answer. Jude? Yeah.
No, if I understand your question correctly, it hinges on really about how you think about differentiation, correct? Because at the end of the day, the way we tend to, again, if you're in a space where others are, or a space you're already in and others are coming in, the next generation products that we focus on really elevating the true differentiation. And it isn't always about potency. It isn't always about half-life. You sort of have to study the liabilities of what the dominant player is in the market and over-deliver on a differentiated product. There are key parameters you focus: performance, which is efficacy or safety or balance of the two. The second is convenience. You know, if convenience is really a true differentiator, you can over-deliver on the convenience factor. The third piece is, frankly, cost. You can differentiate significantly on cost. And the last is, of course, novelty and patentability. You want to make sure that it's not only differentiated, but it's also novel, and one that you can have a good patent position for a good time.
I see the pizza's there, so
I just want to just thank you for coming, and just make the point that
if we want San Diego to be great, you know, we want to attract the best and brightest, we have to have a vibrant interactions between pharma and academia. And so Neurocrine is a, you know, fantastic partner in that, and that's going to help build our future. So we want to embrace all that Neurocrine and other companies like it have to offer. And I hope that you take the opportunity to write Jerold's email address down and contact Neurocrine for questions or opportunities or collaborations or anything that comes up. And thank you all for participating today.
yeah, thank you. And thank everyone online as well.